Thank you for joining us for this second session of Investing in Innovation. I'm also preparing for my end of the year board meeting. And looking at gathering some data, it looks like all of the investment trends are pointing the wrong way. They are down from seed to A round. And the life science sector was especially hit very hard. Therefore, the tools that we're highlighting today, the tools in which universities are investing in promising science to allow it to become a financeable opportunity, are important more than ever before. We're very happy to partner with Michigan and Yale in hosting this webinar where we're going to highlight some of our most promising technologies, where we invested our own resources and transformed them from basic science to something that should and will become a startup company or a licensable opportunity. With that, I will allow Kelly to continue their introductory remarks.
Thank you, Amir. Good afternoon. I'm Kelly Sexton, Associate Vice President for Research, Innovation Partnerships, and Economic Impact at the University of Michigan. In this role, I oversee Innovation Partnerships, which is the University of Michigan's research commercialization arm. As Amir said, our institutions recognize that we must invest time and energy to move our innovations into the world. We are very fortunate to have translational funding and business mentorship as resources that we can put to use to help advance and de-risk our technologies. We are so excited to collaborate with UCLA's Technology Development Group and with Yale Ventures to create a new avenue for life science investors to readily connect with some of the most promising life science opportunities from our 3 prestigious academic medical centers. Thank you to our academic presenters, and most especially thank you to the investors that are joining this session today. I'm looking forward to the presentations.
Good afternoon, everyone. This is Josh Jabal. I'm the Managing Director of Yale Ventures, sending greetings from New Haven, the pizza capital of the universe. And just simply want to add my thanks to Amir and the team at UCLA for putting on this session, and for all the, and of course, Kelly. Also, congratulations, Kelly and our Michigan colleagues, for a great win last weekend against Ohio State. Say that as the son of a Michigan alum, very happy for that outcome. But really thank you to our investigators presenting today and the investors who've joined. Just put in a quick pitch for any of the investors who are planning to come to Pitch Fest, which is one of our second biggest event of the year, which is next week, next Thursday in New Haven, where 32 of our investigators will be presenting really the next phase of new technologies emerging from our labs. If any of you are coming to that, we're having an investor-only dinner the night before on Wednesday. If you're interested in that, please send me an email or ping me in the chat here. We'd be delighted to have you. But thanks again for joining tonight.
Thank you, everyone. And I'm Marvi Valcourt, the Associate Director of Marketing and Communications for UCLA TDG. I will be helping to moderate the event today, and we have several PIs that will be presenting today. The format of today will be 8 minutes of presentation and 2 minutes of question and answer. You can ask your questions in the chat or in the Q&A module. I have a timer that's going to start, and so Isabel, if you would like to start, we can get going.
Thank you, Marvi, for this opportunity. All right, I'm excited to pitch to you all our non-viral gene therapy approach to treat xerostomia. And so I'm an associate professor with more than 17 years of experience in the salivary gland space, and I'm assisted by a phenomenal team of experts with expertise in a variety of disciplines to commercialize and translate our product. And so we're really focused on more than 75,000 patients that are diagnosed in the US each year with head and neck cancer. And all of these patients receive radiotherapy treatment as their primary care, but they also start to develop xerostomia. And the reason for that is that radiation not only eliminates the tumor, but it's also damaging all the healthy tissues in that radiation field. And one of those are the salivary glands. And if you're not really familiar with this organ is producing saliva, which is important for a variety of bodily functions. But it's also a branching organ. And so imagine that you are dealing with a tree, but instead of the leaves, we have grapes that produce saliva. And that saliva gets transported through a tube structure. And most of the organs can repair themselves after radiation damage. But the salivary glands really are unique because they're highly sensitive to radiation. And it's really these grapes that are immediately destroyed by radiation. And so all you're left with really is a tube structure in that organ that is water impermeable and therefore immediately stops functioning. And so what does that mean for those patients? Well, immediately during radiotherapy treatment, they start suffering from xerostomia or dry mouth. And you can see some clinical implications here on these pictures, but for most of us, it's really hard to understand what that is. So imagine that you were sick and you have a little bit of a dry mouth, multiply that 100 times, implement excruciating pain, decaying teeth, as well as anything that you put in your mouth becomes like sandpaper because you've lost your taste. And so you lose weight and you get a lot of other health issues. So those are patients that finally survive their cancer, but are now dealing with a reduced quality of life for the rest of their lifetime. And so to make matters worse, there's really no treatment on the market right now. Those patients, you can easily identify them because they have a bottle of water in their hand because they constantly need to lubricate their mouth. There's a small fraction of patients that can be treated by a drug called pilocarpine, but really the physicians don't like to prescribe it because it causes such severe side effects such as nausea and diarrhea. And so one way to help these patients is to really re-engineer that damaged organ. And a dear colleague of mine years ago came up with the idea to introduce a water channel into that remaining tube structure. And what that does is it's going to release water through the natural process of osmosis. And actually, this concept works because it's been shown to be effective in phase 1 clinical trials already. First, they used an adenovirus, but unfortunately, that never progressed because there is a strong immune response against that viral vector. In a second attempt, they used an adeno-associated virus, and even though that is better than an adenovirus, it's still very immune provocative. And so that means that a reapplication of this therapy may not be feasible. And at one point that will be needed because these tube structures replace themselves over time and no longer have that water channel then, and also a viral vector only lives that long. And so we're proposing a non-viral gene therapy approach, but we have to alter a little bit the biophysics of the cell by which we can bring that vector into the cell, because right now, foreign DNA is only taken up by the cell in 2 scenarios. One is at the time of conception, and the second one is through viruses. But we're dealing with a non-viral vector, so we're going to bring in ultrasound and microbubbles. So how is this going to work? Well, the patient comes in, opens their mouth. We can easily see the opening of the tube structure. We are going to place a catheter in there and then infuse a fluid that consists of the non-viral vector and the microbubbles. Then we're applying ultrasound over the skin that overlies the gland. And what this does is it's going to oscillate the microbubbles, which temporarily open the membrane. The plasmid enters the cell and we get expression of the water channel. And the good thing is we can apply this as many times as we want. For the patient, this is fairly quickly. They sit in the clinical chair for 30 minutes and then they easily can go home. Now, the technique itself is not new because retrograde infusion of fluids into the gland has been done for decades, and also the use of ultrasound for glands has been clinically established. But what we infuse, that is entirely new. And so for the regulatory path, we've already established that this requires an IND-supported BLA. It's important to note that both the microbubbles and the ultrasound device of our investigational product are already FDA approved and cleared. And so for protection, we are going to request an orphan disease market exclusivity, as well as a reference product exclusivity. And our product can also serve as a technology platform in the future for other gene therapies that already exist or other diseases. And so really, what is our opportunity? Well, we can easily go up in the billions of dollars. We will start with cancer specialty centers, but then open it entirely up, not only to radiation-induced xerostomia patients, but also those that develop xerostomia due to autoimmune diseases. And so who are our competitors? As I mentioned earlier, the current standard of care isn't really a competitor, but we are looking at the phase 1 trial of the AAV aquaporin 1. As I mentioned, this may only be applied one time. And in the recent years, also cell therapy entered phase 1, but we don't know yet whether that will provide long-term treatment. And so we really see our investigational product as superior because it can be reapplied multiple times to provide long-term treatment. And it can be used as a standalone treatment, but also with existing therapies. And so we have over 13 years of research data already. We've been very fortunate to receive funding from the NIH for over $3 million already. And so we're really seeking investment to achieve our IND submission over the next 24 months by completing a GLP toxicology study for which we already received partial funding through a translational competitive fund. We also need to perform a vector biodistribution assay, which is necessary to demonstrate to the FDA that our vector is not migrating to other organs in the body. And then simultaneously, but definitely independently, we are also aiming to perform a pharmacodynamic and kinetic study so that this can inform our clinical design and this can be implemented in our IND package. And of course, we want to go all the way to our proof of concept with a first-in-human trial. We already defined 2 clinical sites so that we can generate proof of concept data at a 6-month and a 12-month time point. And with that, I am more than happy to answer questions.
Thank you, Isabel, for that presentation. If you have questions, again, you can enter it in the chat or the Q&A function. So we'll give it a few seconds to see if anyone has questions. One question here. Is administration done under only local/topical anesthesia?
Yes, that is a great question. Most ENTs who will be providing this can actually do it without local anesthesia, but some will could prefer it. At least that is what we gathered out of our market assessments and other customary assessments.
Okay, next question. Have you explored microfluidic, sorry if I mispronounced that, microfluidic approaches to introduce the viral vectors?
Microfluidic approaches, no. I think it's a great idea, but we have not approached that. But I'm happy to chat about it and brainstorm and get this going.
Next question. Do you have IP to do this with other targets, NKCC1 or CLCS?
Yes, also a fantastic question. For others, we don't have IP yet, but we know that with additional studies and data, we could obtain that, yes.
Next question. Dry mouth is a function of chemo in general. Are you targeting only certain treatments?
Yes, so that is another set of patients that we could potentially target. Right now, we're really focused on the radiation-induced one, but as I mentioned, we can open this up to other patients that suffer from this, absolutely. And to actually add to that, for instance, the autoimmune-induced xerostomia patients, they also have issues with their lacrimal glands, and so that could be another avenue.
Okay, and final question: what is the minimum number of times to this treatment has to be repeated for it to be effective?
Yes, also an excellent question. So that really will have to come out from the first-in-human trial. From the animal studies, our non-viral vector approach gives a gene expression quite similar to that has been seen with the viral approaches. But we do know that the human situation is not always the same as what we see in animals, so we're really going to have to wait and see. But of course, with the beauty of our approach is that whenever a patient starts complaining or suffering again from xerostomia, we can easily reapply, and that is not so much the case with viral-based approaches.
Great. Thank you so much again, Isabel. We appreciate it.
You're welcome. If there's any more questions, feel free to reach out.
Next up, we have Yale's Mustafa Khokha. I hope I said that correctly. Thank you so much. And you can start whenever you're ready.
I'm Mustafa Khokha. I'm a professor of pediatrics at Yale. I'm actually a developmental biologist. I'm a pediatric critical care physician that takes care of children with birth defects. We sequence children and identify genes that might cause their birth defects, and then we study those birth defects in the lab. And so one of my major interests is developmental biology. And one of those interests, for example, is this striking result that if we overexpress beta-catenin or Wnt signaling, we can make these two-headed frog embryos. And so from a developmental biology standpoint, Wnt signaling is very interesting to me. And as part of that, we've made rather a striking discovery about Wnt signaling, which I think has real opportunity for cancer therapy. So Wnt signaling actually is one of the biggest causes of various cancers, including colon, breast, lung, multiple hematopoietic malignancies. In fact, if you just consider colon cancer, we're talking about 832,000 deaths per year with a $10 billion therapeutic market. And 90% of these colorectal cancers are due to excessive amounts of Wnt signaling. And if you look at the Wnt signaling pathway, one of the key parts to this pathway is for beta-catenin to go into the nucleus and activate a program of cell growth. And so most of the mutations lead to mutations in genes that are actually degrading beta-catenin and preventing its nuclear entry. And so one of the big therapeutic questions is that if we could prevent beta-catenin from going into the nucleus, we could have a potential therapy. And in fact, other therapeutic strategies potentially activating these has not been very effective. And so there really isn't a therapy here. And of course, the reason why there isn't a therapy for nuclear transport of beta-catenin is we simply don't know how beta-catenin enters the nucleus. And so if we could block it, we could block cell growth and have a therapeutic therapeutic avenue for treatment of these patients. And so in order to understand nuclear transport, there's three things that I need to tell you about. I need to tell you that nuclear transport is dependent on RAN. It's dependent on the cargo having a nuclear localization signal. And it's key that it has a nuclear transport receptor, in this case, important as I've described here. And if you look at beta-catenin, we don't know that it's RAN dependent. We don't know that what its nuclear localization signal is, and we don't know what its nuclear transport receptor is. And that's because beta-catenin is thought to go into the nucleus sort of autonomously. And in fact, what we've shown now is that that is not true. That in fact, beta-catenin nuclear entry is RAN dependent. It does have a particular nuclear localization signal, and that it uses a nuclear transport receptor, transportin 1, 2. And this is really our major advance, really sort of transforming the field of beta-catenin. And what's particularly striking is that the nuclear localization signal really requires 2 amino acids to be functional. And so we think by targeting just these 2 amino acids, we can potentially inhibit its function. In fact, we even have a proof of principle, which is that if we take, for example, this is just to show that we can actually show that if we modify these 2 residues, we can completely eliminate binding of beta-catenin to transportin 1. And I think what I'd really like to point out here is that if we use a chemical inhibitor, you can imagine here, we could prevent the nuclear entry of beta-catenin. And so that's really the goal here. And to do that, we've actually found a peptide inhibitor as a proof of principle. So there's a well-known peptide that binds to transportin very tightly. Our hope was to have that competitively inhibit with beta-catenin, and therefore prevent its nuclear entry. And you can see that as we add this peptide, activation of the Wnt signaling pathway is significantly reduced, suggesting that there is a proof of principle that beta-catenin can be inhibited. Beta-catenin's nuclear entry can be inhibited by an exogenous peptide or small molecule. And so that has been the goal of our work so far. We've gotten secured Blavatnik funding here at Yale to do a drug discovery strategy. The idea is to screen for small molecule inhibitors rather than a peptide, and then do a protein-protein interaction screen to see if any of those prevent the transportin beta-catenin interaction, and then to validate those either in mouse cell lines and then in vivo, potentially in xenopus, which we have a lot of experience in. And once we have a drug that looks exciting, we could then sort of optimize it for IND-enabling studies. And so where we are at right now is we've done a virtual chemical screen looking at that specific region of beta-catenin that's really critical for function. And we've looked in the large space, screening basically 6 million compounds. We've then used a number of different filtering strategy and have come down to about 300 compounds that we are very excited about. Those compounds are interesting in the sense that many of them are have a wide variety of different aspects to them as far as ion charge and other chemical compositions. And so we think that there's a lot of space that we could potentially test. And actually some of them are fragment-like because the region is small, which then offers the opportunity to also do IND studies to make them potential therapies. And so that's where we are right now. And of course, certainly we're looking for funding to expand this. The more compounds we can screen, of course, the more likely we are to find something that can inhibit that interaction between transportin and beta-catenin. And so we're certainly looking for partners to help us with these screens. Okay, I think that's all I've got. Happy to take questions.
All right. Okay, here's our first question. My question is about the competitive landscape. Why is a small molecule inhibitor of the pathway better than an antibody approach such as Amgen drugs, the clinic targeting this pathway?
Yeah, so I mean, I think, you know, an antibody is a potential possibility. I guess we were thinking about something that would work intracellularly. And so, you know, in order to prevent nuclear entry of beta-catenin, in which case a small molecule may be more suitable for penetrating through at least one layer of membrane. And so that's why we sort of focused on a small molecule rather than an antibody. That's also a possibility, I suppose. I think we thought we'd start with the small molecules first as a strategy.
Also, what is the drug profile like? Peptide drugs are notoriously unstable. Is it possible to explore med chemical principles to occupy the active site with a traditional drug-like molecule?
Yeah, so that's exactly right. So the peptide was mainly, our goal with the peptide was mainly just to show proof of principle to sort of just, you know, because there's well-known peptides that bind to transportin that are known nuclear localization signals. So testing that was really sort of our proof of principle. But we totally agree with you. Our goal is now to try to find a small molecule, a chemical, not a peptide, for the reasons that you just described that we could use therapeutically. Yeah, that's exactly the goal here for this, especially for this high throughput virtual screen. And those compounds now are in hand and we're in the process of screening them.
Based on the experience with cancrese inhibitors demonstrating on-target toxicity in GI crypt cells, how do you imagine achieving progressible TI without a mutant selective approach?
Mutant selective approach. Maybe could I ask you to clarify your question? Is your concern toxicity? I mean, sir, certainly toxicity is a concern. Certainly in the crypt cells, Wnt signaling is important for maintaining it. But if you look at the Wnt signaling that's occurring in cancer cells, they are really off charts. They're really off target. So our hope is that by simply reducing that level of Wnt signaling, we can find a therapeutic window between excessive growth and slowing down that excessive growth and normal growth, which is necessary. And again, we're looking at cancer here where the therapeutic windows tend to be considerably narrower than other disease types.
Next question. How are you screening for specificity versus other transportin cargos? Correct.
So the key there is to actually target beta-catenin. So you're right. If we were to target transportin itself, there are going to be a number of different cargos that we would inhibit. This, we're looking for a specific target on beta-catenin that would bind to beta-catenin and prevent its interaction, not binding to transportin. And that's how this virtual screen has been designed.
Next question. What is the ask? How much are you raising?
What is the ask? That's a good question. I think what we'd like to do is, as a first step, is to raise enough funds to do this screen. I think we're looking at around $250,000, so it's a relatively small ask for this first step.
So the follow-up to the question before, what is the effect of the Win knockout experiment? Is this target a viable candidate for a degrader molecule, such as those developed in Craig Crew's lab?
Yeah. So that's an interesting question. Could we actually make a degrader molecule for beta-catenin? Yeah, I think those are also possible. I think the challenge there is to be able to label beta-catenin in a successful way, or in a way that's specific to just the cancer cells. And I think that's where the challenge might be in some of these degrader sort of strategies. Again, so the option would be to label all molecules, which of course would be really challenging in a whole body, or to find a small molecule that could block the signaling, again, hoping to target those cells that have excessively high beta-catenin signaling, like you might have with the cancer cells. And we thought there a small molecule would be most effective.
Great, thank you so much. We are going to move on to Gloria Huang, also from Yale. Welcome, Gloria.
Thank you so much for the opportunity to present our work today. Today I'll be talking about a new precision medicine for ARID1A mutated ovarian and uterine cancer. My name is Gloria Huang. I'm an associate professor at Yale, and I have many years of experience leading a translational research lab and being at the forefront of designing clinical trials to advance and change the practice of gynecologic oncology. In this current project, I'm partnering with Yale Center for Molecular Discovery, and I'm supported by Yale Ventures and the business development team. The problem that our team is addressing is that ovarian cancer patients lack targeted and effective therapies. The standard of care for advanced ovarian cancer remains surgery and chemotherapy. Although many patients have a good initial response, despite advances in the provision of surgery and advances in chemotherapy, still, with the standard of care, about 80% of patients with advanced ovarian cancer relapse with chemo-resistant disease, which is not curable. Novel therapies for ovarian cancer are limited, and therapies such as PARP inhibitors are not as effective for ARID1A mutated cancers. So specifically, ARID1A mutated cancers represent about 10% of ovarian cancers, and these are mostly clear cell and endometrioid type of cancers. We aim to develop the first targeted therapeutic option for clear cell and endometrioid carcinoma patients with ARID1A mutation. In my lab, we discovered a new function of ARID1A, which is as a regulator of the de novo pyrimidine synthesis pathway, shown on the left side of the figure. This pathway is important for the synthesis of pyrimidine nucleotides for transcription and replication, and is particularly important in rapidly proliferating cells. Other cells can rely primarily on the salvage pathway. As a result of this, ARID1A mutated tumors have hyperactivated pyrimidine synthesis and are hypersensitive and vulnerable to pyrimidine synthesis blockade. We showed this in a variety of approaches, for example, using a repurposed pyrimidine synthesis inhibitor, teriflunomide, and testing this compound in clear cell ovarian cancer cells, which are isogenic paired ARID1A proficient or deficient, and using genetic engineering approaches. As you can see on this in vivo experiment shown on the right side of the slide, in the red are the ARID1A deficient ovarian cancer tumor xenografts, where the growth is effectively suppressed by a low dose of teriflunomide, whereas ARID1A proficient cells are not affected by this drug and are similar to the vehicle treatment. In terms of the potential to directly target CAD, I'll just back up to show the pathway. The CAD enzymes shown on the red boxes. The first 3 enzymes of the de novo pyrimidine synthesis pathway are encoded by the CAD gene. So we validated CAD as a selective therapeutic target for ARID1A mutated cancer using siRNA, which specifically knocked down the CAD gene and resulted in abolishment of the CAD protein expression, as shown by immunoblotting in the middle panel. As expected, the knockdown of CAD was able to reverse the de novo pyrimidine synthesis and decrease the pyrimidine nucleotide intracellular levels. Shown on the right is the differential effect of CAD knockdown on cell proliferation and viability is dependent on the ARID1A status. So in the paired shown in both colorectal cancer cell line HCT116 and a clear cell ovarian cancer ES2, the cell viability is markedly diminished when CAD is knocked down, specifically in the ARID1A deficient cells. We have the advantage of being able to learn from previous attempts to inhibit CAD, specifically the aspartate transcarbamylase enzyme of CAD, encoded by CAD in the clinic. And this was done through a compound PALA, which is an analog that is an inhibitor of the aspartate transcarbamylase. It was historically tested, however, was not successful in advancing to the clinic, in part due to suboptimal activity of the compound, which was found to have actually activating activity at low concentrations, and was not tested in tumor types which have a high rate of ARID1A mutation. And our approach for clinical development will be informed by this historical experience by selecting patients for ARID1A mutation, which we have readily available tools, and by choosing the correct population as well as synergistic drug combinations. This will increase the success of our precision medicine strategy. We have undertaken work to identify and evaluate novel CAD aspartate transcarbamylase inhibitors, and this was done through modeling of the protein-protein interaction sites of the ARID1A and CAD aspartate transcarbamylase, and then virtual screening of a very large unique compound library. And we are undergoing cell-based testing of the compounds using our unique preclinical cell models with isogenic paired ARID1A mutated and wild type cancer cells. And already just in testing the initial hits, we have already identified compounds that have over 10-fold improved potency as well as selectivity for the ARID1A deficient cells compared to the historical compound of PALA. However, we are still underway testing additional hits as well as additional small molecule library screen. Just to again mention that this would be developed in a biomarker-directed therapy with a strategic partnership to develop a tumor DNA companion diagnostic test. And this is something that ARID1A mutation can be detected at very high sensitivity, both in tumor tissue as well as in the circulating tumor DNA. In terms of the potential market, as I mentioned, 10% of ovarian cancers are ARID1A mutated and lack effective therapy. And in addition, uterine cancer, which is a very common cancer in the United States, 1/3 of uterine cancers are ARID1A mutated. And the global market for ARID1A mutated gynecological cancers is estimated to be $1 billion. And market drivers include the increasing incidence of ARID1A driven cancers, increased genetic understanding that can underpin the development of targeted therapies. In terms of the competitive landscape for ARID1A ovarian cancer therapies, we believe that the CAD inhibitor that we're developing has greater first-in-line potential and greater personalization targeting ARID1A mutation compared to the other agents that are either FDA approved or in development. For example, phase 2 evaluations of EZH2 inhibitors and combination DNA damage repair inhibitors. In terms of the discovery process and future plans, our prior work was supported by the DoD Ovarian Cancer Research Program, and the in silico screen and the current cell-based screens have been supported by the Blavatnik Fund for Innovation. And as we are building momentum to bring this precision medicine strategy to the clinic, we are seeking additional funding to support and accelerate this goal. I've included my contact information, my email address, and I would be delighted to be contacted, and as well as the email address for the Director of Business Development.
Okay, so we have some time for questions. And again, if you can enter it into the Q&A module or in chat, I'd be happy to verbalize these. So Gloria, the first question, any ARID1A organ system targets beyond ovarian and uterine cancer?
Yes, that's a great question. So ARID1A mutations are actually, ARID1A is one of the most commonly mutated genes in human cancer, and 5 to 10% of all human cancers have an ARID1A mutation. And I have done testing of utilizing isogenic paired non-GYN cancers, such as colorectal cancer, with showing that same therapeutic selectivity. And some of the other cancers that have high rates of ARID1A mutations include liver cancer, gastric cancer, and others.
Thank you. Okay, are there any other questions for Gloria? Any potentially off-target safety issues?
So interestingly, de novo pyrimidine synthesis inhibitors are quite safe, and actually I showed very briefly data using a repurposed compound which targets the enzyme immediately downstream of CAD, which is the DHODH, and there are 2 DHODH inhibitors that are FDA approved for non-oncological purposes. They're actually anti-rheumatic. One is used for multiple sclerosis and one for rheumatoid arthritis, and actually are quite well tolerated for extended daily oral use. The CAD inhibitors actually have a minimal effect on ARID1A proficient cells, and actually the cellular effects are very different between the ARID1A proficient and deficient cells. There's definitely some profound differences between the ARID1A proficient and deficient cells and their response to these inhibitors.
Great, thank you so much, Gloria. We really appreciate it.
Thank you.
So we are going to move on. So next up is UCLA PIs, and the first one will be Nalo Hamilton, and you can start whenever you are comfortable.
Good afternoon, everyone. My name is Nalo Hamilton. I'm here from UCLA. I am an associate professor here in the UCLA School of Nursing. And today I'm going to talk to you about our development of novel inhibitors for the prevention of triple-negative breast cancer progression. We are looking at triple-negative breast cancer because we need to understand the biological differences that happen with this aggressive cancer. It accounts for only 15% of breast cancer. So if the annual occurrence is 240,000, then it would mean only about 36,000 women are diagnosed with breast cancer. However, of the 42,000 women who die annually of breast cancer, 50% of them are due to those who have triple-negative breast cancer diagnoses. And that is because there is a lack of targeted therapies for this type of cancer. Our team has developed novel and targeted small molecules for triple-negative breast cancer, and we hope that it will help for women not only in later stages, but primarily in earlier stages of development of cancer. And so we are in the developmental stage of testing these molecules. Our team consists of myself. I'm a biochemist as well as a nurse practitioner, along with other physicians who have been in the development of currently used molecules to treat such things as prostate cancer, as well as Receptor, which is breast cancer. And then we also have colleagues who are pathologists as well and biostatisticians. Now, the interesting thing about triple-negative breast cancer that frequently occurs among premenopausal women, up until May of this year, premenopausal women were largely not screened for breast cancer. And so that means women less than the age of 45 years of age had challenges in determining what type of breast cancer that they had and getting access to that. And the majority of them largely had triple-negative breast cancer. The reason why we're concerned about this is, one, is that triple-negative breast cancer is very aggressive. So there is a high incidence of distant metastases. And as we can see here from the SEER database, is that distant metastases in 5 years, the relative survival is only 12% of women with this diagnosis. So we're looking at a molecule that is the insulin-like growth factor 2, or IGF-2. The reason why we're looking at that, similar to IGF-1, which has also been tested. IGF-2 we have found is highly expressed in triple-negative breast cancer for women who are of African American and Latina descent. The other thing we've also noticed about IGF-2 is IGF-2 is turned on not only with placental development, but it's also turned on at breast budding. It's turned on with lactation, then it turns off, and then it turns on again with breast cancer. And what we've noticed that IGF-2, like IGF-1, when it binds to its receptors, it has a signaling that downstream allows for cell proliferation and growth, protein synthesis, migration, which is a huge concern, especially with triple-negative breast cancer, and also some changes in some metabolic effects. Now, we've developed some small molecules at this time that are able to inhibit IGF-2 signaling. And we hope that this will develop into long-term therapeutic options that we'll be able to use alone and in combined therapies. We know that with cancer, the best outcomes we have are when we use in combined therapies. Sometimes there are those that respond really well to single therapies. And so we are encouraged by this because, as I mentioned before, the USPSTF indicated that starting in May that we will now start screening women for breast cancer in their 40s, which is a change because by and large, screening was supported for breast cancer in women who were 50 years of age and older. And so we anticipate, similar to what we see here, is that we're going to see an increase, not only in the detection of breast cancer, but also in triple-negative breast cancer. And what this shows us here is that we have an incident in breast cancer that is specifically triple-negative, women who are African-American across all age groups, starting in their 20s up until the age of 45 to 49. And then we have an increase followed by those who are Caucasian and who are Hispanic. Again, as I mentioned, TNBC largely affects women who are premenopausal. And so it is possible that IGF-2 is actually an indicator and a target for early development of breast cancer. So you might ask about the competitive landscape. I have that listed here. We have several therapies that are offered now for triple negative breast cancer treatment. And we have those that are offered by AstraZeneca, but that's only for individuals who have various mutations who will respond to PARP inhibitors. We do have Trodelvy, but that is for women who have metastatic triple negative breast cancer and have already failed 2 therapies. Our small molecules are being designed to help with women who have early onset or smaller tumors of TNBC and have not had distant metastases yet. We also have Keytruda that's available currently, and that is for women who have tumors that have an overexpression of PD-1. And so there are only a small minority of TNBC patients that could benefit from that. And so with the fact that IGF-2 is expressed, we have published in our team that we see an increase of IGF-2 expressed in TNBC, both in premenopausal women with breast cancer, premenopausal women diagnosed with TNBC, as well as those with aggressive ER-negative breast cancer. Excuse me, I messed up. So here's my budget of milestones. Currently, we have tested several of our analogs using a panel of TNBC cell lines. We are also using some of 2 of them right now that we have tested in vivo, but we still need to do the pharmacokinetics, because with previous IGF-1R inhibitors, they have had some off-target effects. And so we are thinking that our formulation will do better without having those off-target effects, which will allow us to have better sustainability and better ability to combine our small molecules with those that are currently available and being used now in therapy. We know that we're going to have to do some cytokine assays that could tend to be a little expensive, as well as using our single and combination therapies. We also want to do immunohistochemistry, and we'd also like to use PDX models as well. So I want to thank you for your time and attention, and hopefully I've sparked some interest in some collaboration, and I'm ready for questions.
Okay, Nalo. First question, is TNBC a factor for male breast cancer?
Actually, you do have males who do get triple negative breast cancer. On average, you have 2,100 males who have triple negative breast cancer. And then of those number, about 500 males die annually. The concern we have with triple negative breast cancer is it doesn't have the markers that we need to treat with our current therapy. So they lack the expression of the estrogen receptor, the progesterone receptor in the nucleus, and they lack the overexpression of HER2. So the current therapies that we use that allow patients to have longevity, increase their long-term survival, are basically targeting those types of receptors. We don't have that type of target for those who have triple negative breast cancer, female or male.
Okay, here's the next question.
Okay.
How are you different from past LGFR attempts that have a sorted set of results and no big win?
Okay, love the question. I love that, it helps with the skepticism. But see, I think one of the challenges that we also have with this is that we're not screening cancers for the overexpression of IGF-2. So the interesting thing about our lab and our approach is that we're looking not only at the tumors, so we need to screen them first for IGF-2 to hope that they have the response. And the other part is also developing the therapies that will directly target that. The other thing that we have with my general skeptic is that we're also trying to develop molecules that don't have those off-target effects. We can't always know what those are, because I think that in most of the tests that we've done up until this point, we haven't also looked at the cytokine profile. And that might be something that we need to adjust. We do have other therapies that are out there. We are using one as a parent model, but we have also found in our studies and in my lab that there might be some the molecules that are being recruited to the tumor that not only lead to the decrease or diminished size of the tumor, but they are also regulating the immune system. And somehow, if we could harness that based through structures of our molecule, that will be able to have a better outcome. And so we could have more wins than what has happened in the past.
Thank you so much, Nalo. We're going to move on. And Matthew Rettig, if you could turn on your camera and you can start whenever you are comfortable.
Okay, hello, good afternoon. Thanks for the opportunity to present our work. My name is Matt Rettig. I'm a professor of medicine urology at UCLA. I'm the medical director of the prostate cancer program. And today I'll be telling you about inhibitors of the androgen receptor, the AR, N-terminal transactivation domain, predominantly for the treatment of advanced prostate cancer. So here are the key team members, myself, really focusing on the biology and clinical applications of our compounds, and Mike Jung, the medicinal chemist who has extensive history in prostate cancer as the co-inventor of both Xtandi and Erleada, which are major commercial successes in this space. So prostate cancer is the second most common cause of cancer-related deaths amongst men in the US. It's the #1 killer of men who are non-smokers, and it has the highest incidence of any malignancy, with approximately 270,000 cases expected in 2023 and about 35,000 deaths. The number of deaths has been increasing year over year since 2018. Currently, in the clinic, all of the compounds either directly or indirectly target the ligand binding domain of the androgen receptor. That is, the domain of the androgen receptor that interacts with androgen. All of these compounds are associated with drug resistance that is mediated in large part through the ligand binding domain. So to overcome this issue, this drug resistance, the ideal compound drug would target the N-terminus of the androgen receptor. Importantly, in prostate cancer, even when tumors are resistant to androgen deprivation therapies and the novel hormone signaling agents, the tumors are largely still dependent upon the androgen receptor for survival and proliferation. So the development of compounds that target the N-terminus has been a challenge in general because the N-terminal domain does not have a known structure. It's called an intrinsically disordered domain. So structure-based drug design is not feasible. Our ideal compound, we feel, would be one that targets the N-terminus and does so through degradation of the androgen receptor. We undertook a high-throughput drug screen through the NCI library, and we did this in yeast and identified a lead compound, confirmed its relative selectivity for the androgen receptor, and have done extensive SAR, developing approximately 350 analogs. And I'll tell you about one of the principal leads, although we have several backups that are currently in further testing and validation. So what we're seeing here is the selectivity of our lead JN103 for inhibiting the growth in vitro of AR expressing but not AR null prostate cancer cells. On the right, what we see is the induction of AR degradation by our compound JN103, but not other closely related nuclear steroid receptors such as the glucocorticoid receptor, estrogen or progesterone receptor. Here I'm showing you the selectivity of our compounds. Here there are approximately 20 different cancer cell lines. And in this in vitro growth assay, we see that none of them are affected by our compound, JN103. The one cell line that is affected is a breast cancer cell line that actually expresses the AR and is dependent upon the AR for its survival and growth. Importantly, we have shown that our compound covalently binds to the androgen receptor and does so through an interaction with cysteine at position 178, and that has been demonstrated through mass spec and functional mutagenesis assays. In vivo, our compounds have activity when administered orally. This particular model shown here, 22RV1, is a hyperaggressive castration-resistant model, and we've grown the tumors in mice that have been castrated, and once tumors reach 200 cubic millimeters, they're then treated with our compound. Our compound, JN103, is shown in yellow. Enzalutamide, the blockbuster that was developed by Mike Jung and colleagues, is shown in orange. So you can clearly see that our compound has good activity in this aggressive castration-resistant model. And this slide is showing the pharmacodynamic effects of our compounds targeting the AR in vivo. On the top are the Western blots, and on the bottom are immunohistochemistry stains demonstrating reduction in AR expression. So I don't know that I need to tell this audience about the market opportunity for prostate cancer. It's really a massive market, both in the US and globally. In terms of the competitive landscape, there are many compounds that are currently on the market, all of which target the ligand binding domain. And the resistance to these compounds largely are driven by alterations in AR and AR signaling, as opposed to AR independent or bypass mechanisms. And our compounds overcome these resistance mechanisms in large part by targeting the N-terminal domain of the AR and inducing its rapid degradation through covalent binding to, as I mentioned, a specific cysteine residue in the N-terminus of the AR. So in terms of risks and mitigations, first of all, I should point out that I'm just sort of scratching the surface on what has been done. We have tested our compounds in many in vivo models and have done some initial in vitro and in vivo PK. We have 5 compounds that have similar or improved in vitro activity, some of which have already been tested for PK and have improved PK as compared to JN103, which has been the focus of this talk. Of course, we have to do extensive ADME tox to demonstrate the safety and potential off-target effects. But to date, we haven't seen any in vivo toxicity at the highest dose tested, which is 40 milligrams per kilogram once daily, based on mouse behavior, weight, or blood tests. So we think our compounds have a real potential for activity in the advanced prostate cancer space. So I want to thank you for listening, and hope that there's interest, and would be happy to field any questions that you may have.
Thank you, Matthew. Question. Sorry, I might have missed the relevant slide, but what is the selectivity of the inhibitor, and how much of a concern is off-target binding and degradation?
Yeah, so it's highly selective. So we in addition to just demonstrating the selective degradation of nuclear steroid receptors, we've done proteomics screens. So basically, we take our compound and see the effects on protein expression in an AR null prostate cancer cell line. We actually see absolutely no reduction in any protein in a proteomics screen. So we think there's a high degree of selectivity. We have done some cysteine immunoprecipitation assays, and we do see that there are some proteins to which our compound binds, but those proteins do not have their expression level affected by our compound.
How will this differentiate from AR-110 other than covalent versus non-covalent inhibitor?
The AR compounds, the Arvinus compounds, target the ligand binding domain. So that's one critically important distinction. We know that there are important mechanisms of resistance, including the expression of constitutively active variants of the AR that lack a ligand binding domain. So that's one important distinction. In addition, the ARV-110 compound actually only works in a small subset of AR variants, and those that have specific point mutations in the ligand binding domain. And that's not demonstrated just in vitro. That's actually been shown in their early phase clinical trials. So in the development of ARV-110, they're really focusing on about the 5 to 10% of patients who have the specific AR variants that are susceptible to AR-110.
Speaking generally in the context of the advantages of degrader molecules, does using a covalent inhibitor create a disadvantage because you lose the catalytic activity that is normally highlighted by degraders allowing low dosing?
Actually, I think there's some pharmacokinetic advantages with a covalent binder. So I think they're less dependent upon an AUC. They're perhaps a little bit more dependent upon a peak concentration. But with our compound, the one that I've predominantly focused on, JN103, it actually has less than ideal PK features, but it nonetheless has significant pharmacodynamic effects in vivo. And the compounds that we have that we're currently testing to validate in vivo activity seem to have improved PKs compared to JN103. So we think we can improve on the activity of our compound and have excellent anti-tumor effects. There are certainly theoretical issues that you mentioned related to mechanisms of action, but ultimately I think the proof is in the pudding. If it works and works selectively, I think that's the ultimate goal. And I think our compounds are on their way in that regard.
Interesting that you can be better than ENZA in some non-clinical models. Is your vision to be a better than ENZA and ABI and combos to be good after?
Yeah, so in terms of clinical development, it probably makes most sense, at least initially in the first in human, to take those subjects who've been treated and progressed on ENZA and other novel AR signaling inhibitors. In terms of the long-term development, you could imagine that this compound or these compounds could have activity not only in the most advanced stages of disease, but earlier on in castration-resistant space, either alone or in combination with other AR targeting agents or other agents in general. And as we've seen with ENZA and other AR targeting agents, they tend to work even better when they're brought earlier and earlier in the disease space, including in the castration-sensitive prostate cancer patient population.
Great, thank you so much, Matthew. We're going to move on, and next is Peter Clark from UCLA.
Great, so thanks so much for inviting me to present my proposal here. So my name is Peter Clark. I'm an associate professor here at UCLA, and my lab has been working on developing a hexokinase 2 inhibitor to treat hepatocellular carcinoma. So just a quick overview of what I'll tell you today. First, I'll tell you why hepatocellular carcinoma, or what I'll abbreviate HCC, why we're in need of new therapies there. I'll explain why hexokinase 2 or HK2 is a really well-validated target in a large portion of these tumors. I'll explain how we've identified a small molecule that we think can selectively target HK2. And then finally, I'll describe my accomplished and expert team that I've put together to really pursue this project. And quite honestly, why don't I just start with the team, because I actually think that's a real strength of our approach here. It includes myself, who has a lot of expertise in assay development and study metabolism. I've also worked with a number of biotech startups, including this company Trithera, so I kind of understand that ecosystem. We have a high throughput screening consultant with Robert DMSO. Harvey Hirschman has done a lot of work on hexokinase 2 and hepatocellular carcinoma, so really understands the biology of that very clearly. We have Varghese John, a medicinal chemist, who's done a lot of work both in industry and academia to develop drugs, some of which have reached clinical trials. And then Richard Finn, who's a liver cancer doctor. He's actually done a lot of work bringing drugs from preclinical development all the way up to global registration. And so he has a lot of expertise in that area. All right, so why do we need new drugs for hepatocellular carcinoma? Like most cancers, hepatocellular carcinoma, if caught early, can be cured by resection, but unfortunately, again, like most cancers, it's not caught early, in which case you really end up with these systemic therapies. Systemic therapies in this case are usually immunotherapy, and there's really pan-kinase inhibitors. They work for sure, but don't work incredibly well, and you can see that the survival, even for intermediate and for certainly advanced stage patients, is very low. We're talking about certainly lower than 5-year survival rates. And so there's a real need for new therapies here. Where do we think we can improve? Well, we're really excited about developing this inhibitor for hexokinase 2, and I'll explain the biology of that in just a second. But I do think we have clear advantages in the way we're developing this. First off, as I'll show you, we're developing a selective inhibitor here, and so we expect limited off-target effects. Our approach is really orthogonal to what most other people are doing. As I mentioned, most other people are looking at immunotherapy, also kinase inhibitors, protein kinase inhibitors. We're looking at a metabolic enzyme here. We know the patient population we're going after, those with overexpression of this enzyme. That's very different from what's out there already. And finally, it's a little subtle, but we actually can use FDG PET, which is used every day in the clinic to look at cancer patients as a pharmacodynamic biomarker of this drug. And so even before we even think about clinical trials, we're already thinking about biomarkers here. For those who may not remember their high school biology or their college biology here, let me give a quick overview about hexokinases. Hexokinases are really the first rate-limiting enzyme in glycolysis. They convert or catalyze glucose to glucose 6-phosphate. Now, there are 4 different hexokinases in the body. Right now, I'll just focus on 2 of them, 1 and 2. Hexokinase 1 is really ubiquitously expressed throughout the body and mostly every tissue. It's likely required for normal physiology. Hexokinase 2, on the other hand, is a very unusual one. It's really only expressed in cancers and then a very limited number of tissues, and I'll show you some data in a bit that seems to be non-essential for adult physiology. And so just to step back right here, what we already know is that as we develop this inhibitor, we need it to be selective for hexokinase 2 over hexokinase 1. Now, I'll quickly give you a few vignettes to show you why the biology of this all works, how we show that HK2 is important for HCC, that was non-essential in adult physiology, and then some human data supporting all this. And kind of the bottom line is work by us and honestly by a lot of other groups have really demonstrated that HK2 is an important target in HCC and one that can be targeted without causing significant toxicities. So, there's lots of data out there showing HK2 is an important enzyme in HCC. Here, I'm just showing you one data point here from Harvey Hirschman's lab. So here, what they did, they developed tumors. They grew up tumors of hepatocellular carcinoma cell lines in mice as xenografts. They then had a genetic construct in these cells, where if you added in the drug doxycycline, you induce an shRNA and block HK2 activity. And hopefully what we can see here is that in those tumors where they decreased HK2 expression, they grew less. Now you may be thinking the effect size here is not incredibly large, and on one hand it isn't. On the other hand, if you compare it to most other therapies in HCC and in other cancers that have reached clinic and had clinical benefit, they kind of have the same effect size. I'll also note that, we can talk about during the questions if you'd like, but there are combination therapies that have already been developed and tested that will combine with HK2 inhibitors that will really block growth entirely here. So that tells us that the HK2 is important for the growth of HCC. This data right here shows that HK2 is not important for at least mouse adult physiology. And so multiple groups have made HK2 knockout mice in adults. And the bottom line is these mice have really no phenotype at all. Homozygous mice have no phenotype whatsoever. They looked at body weight. They looked at fed glucose levels, nothing there. If you look at heterozygous HK2 knockout mice, they have a mild phenotype in terms of decreased glucose consumption in the heart during exercise, but we really don't think that will matter for liver cancer patients. And it's also been shown that HK2 is not required for T cell activation, so we can already think that it can be potentially combined with immunotherapy. That's mouse data. What does the human data tell us? Well, people have looked at HK2 expression in patients, cancer patients, liver cancer patients. And the bottom line is those liver cancer patients that have higher expression of HK2 do much worse than those with lower expression of HK2. And so in our minds, we think if we can take those patients that have a lot of HK2 and knock down the activity, we can really benefit these patients. All right, so that tells us HK2 is a good target. How do we go about doing this? The way we do this is we use a biochemical assay in cells. The idea is we take cells that only express hexokinase 2 as the only enzyme. We treat them with a glucose analog, 2-deoxyglucose. What happens there is that the 2-DG is phosphorylated HK2, and then we convert that into light through a coupled enzyme assay. And the idea here is that any compound that blocks HK2 will block the light, and so we can screen compounds. We then do a counter screen against cells that only express HK1 as the hexokinase, and then we can really identify selective compounds. So what I'll tell you is we screened 70,000 compounds so far, and actually identified a handful that show selectivity for hexokinase 2. What I'll show you here is just one of them, our most exciting one, HK001. And we have data to show, first off, that HK001 can selectively block hexokinase 2 activity in cells. It's selected for hexokinase 2 over hexokinase 1, which is in blue here, that it directly binds to hexokinase 2. We showed that through a thermal shift binding assay, and that it blocks hexokinase 2 activity and in vitro activity in a non-competitive way. So that gives us an early indication that this compound will actually have activity. Now, obviously, there's a competitive landscape out here. We're not the only ones going after HCC, but what I can tell you is that we're really orthogonal and unique in our approach. Again, most people are looking at immunotherapy. Some people are looking at protein tyrosine kinase inhibitors, but really no one else, at least to our knowledge, is developing a hexokinase 2 inhibitor. So finally, where are we going from here? We really have a hit from a high throughput screen, but we need to develop it into a lead drug. And so we need somewhere around 2.5 million to really push forward that medicinal chemistry campaign and test our analogs in vivo. So thank you for listening. I'm really happy to answer any questions, or feel free to reach out to me. Here's my email address if you want to discuss this any further. So thank you.
Thank you so much, Peter.
Thanks.
Okay, so now we have our final presentation of the day is Ariella Shikanov from University of Michigan. Ariella, you can start whenever you're comfortable.
Perfect. Well, thank you so much. I appreciate you all staying that late. The technology that we developed, it's called ARTOVA, and the goal is to restore natural ovarian function for pediatric cancer survivors. If you just take a second to look at these two women, they look similar, maybe related. So these are two identical sisters. They are identical. Clearly, they don't look identical. This picture appeared in the New England Journal of Medicine, demonstrating the effects of chemotherapy on young girls. So the problem starts when young girls before puberty are diagnosed with cancer, and then they receive anti-cancer treatments before puberty. They have a huge risk to develop premature ovarian insufficiency, PO, and they are unable to undergo natural puberty, resulting in significant lifelong complications, such as shorter stature, as you can see here, obesity, high risk of breaking bones, high risk of cardiovascular disease. And right now, we're looking at about 100,000 women who live today with ovarian insufficiency. Unfortunately, there is no treatment. The only available treatment for these girls who cannot start their physiological puberty is hormone replacement therapy, which is off-label indication and suboptimal. It was developed to treat postmenopausal symptoms. HRT, hormonal replacement therapy, delivers only two hormones, estradiol and progesterone, either as a pills or transdermal patches. And these patches, again, are designed for adult women. So young girls who have to use this HRT treatment have to cut these patches in eighths, and then those patches don't stick. If you look at the normal physiological function of ovarian hormones, you can see how different it is from the HRT. So the hormones are fluctuate throughout the months. Estrogen is high in the beginning of the menstrual cycle, and then it drops down. Progesterone picks up later. So it's really dynamic compared to what we can deliver with HRT. So our idea was, what if we take donor ovarian tissue, which is seen here as the pink circles, and then we surround it in a hydrogel that can support tissue growth, and then surround it additionally with immuno-isolating hydrogel, basically creating an immuno-isolating capsule that carries healthy tissue that can respond to circulating hormones and then secrete ovarian hormones in response. Our immuno-isolating capsule can be implanted subcutaneously. It doesn't have to go where ovary is. So it's minimally invasive procedure. It is self-regulating because it's basically healthy, normal cells that respond to circulating hormones. We can use donor ovarian tissue that can, so from one ovary, from a donor, we can make many devices like this. And it's an immuno-isolating capsule that eliminates the need for immune suppression. We also anticipate enhanced patient compliance. So this is how the capsules look like. There's basically transparent capsule with tissue inside. We envision that this capsule can be lifted up and then they can be implanted using a trocar subcutaneously using the same technology as used for Norplant or testosterone pellets. So our team is me. I'm an associate professor at the University of Michigan, and I have 15 plus years working on this. I have an amazing CEO for this company, Kate Remus, and she has 15 plus years in strategy and general management of similar startup companies. Gabby Blevins, she's a master's she's a student who just defended her master's, and she's been working on this project for 3 years now, really leading all the projects. And I have a clinical collaborator, Dr. Molly Moravec, who will be leading the clinical trial, and she's currently the director of the fertility preservation program at the University of Michigan. Since we started working on this project about 8 to 10 years, we've published more than 10 research manuscripts. This research has been supported, so our past funding equals about $3.5 million from the NIH, NSF, and private foundations. And right now I have $7.5 million from the NIH and MTF to continue working on this project for research. We've been awarded a patent, United States patent, earlier this year, and we submitted 2 provisional applications. Right now, the patent is pretty broad. It covers immuno-isolation device for allogeneic and xenogeneic cells for human and veterinarian applications using stem cells, male, female, or genetically engineered cells. We've performed I-Core in 2019, interviewed more than 200 people, physicians, family members, cancer survivors, all supporting our idea and saying that there is actually value for this. So many clinicians said that 50 to 75% of patients don't use HRT as prescribed because it's off-label and it really doesn't fit what these girls need. So we treat this as a one-size-fits-all, and it's not, because we're not talking about postmenopausal symptoms. We're talking about growing body of a growing girl. Also, parents of cancer survivors are really scared to put kids on hormones, so they would prefer something more physiological. We also contracted a Searchlight company that conducted in-depth one-on-one customer discovery interview with medical directors of the US insurance. So here you can see representative from insurance companies. They all agreed that this is an important treatment, and they would pay up to on average $27,000 per year for this capsule. Our market is these young cancer survivors who have to undergo physiological puberty. We're thinking about somewhere between 75 to 100,000 patients right now, plus about 10,000 patients adding every year. If we're being conservative, if we can target a quarter of these people, we're talking about 500 million per year. However, we're also looking to expand. So women who experience premature ovarian insufficiency and postmenopausal women are about close to 20 million people. However, if we're going to expand the market, we will also adjust the pricing. So where we're now, so we completed all the preclinical animal studies with the contract research organization, NAMSA, biocompatibility, acute and long-term systemic toxicity irritation study, passed everything with flying colors. Right now, we are working on submitting our IND, so investigational new drug application, to the FDA. We drafted the phase 1 clinical trial, and we're hoping to submit an IND later by the end of this year, beginning next year. Once we've submitted the IND application, we will start applying for SBIR and VC funding, hoping to raise $10 to $15 million in Series A. We anticipate IND being approved, facilities set up in 2025, and start our clinical trial with the clinical collaborator in 2025, and then expanding to multi-center trials. So why now? The cancer treatments are much better. And as a result, pediatric cancer patient population is increasing and they live very long lives. However, the ovarian toxicity doesn't go anywhere. We've developed a novel cell-based therapy to address an urgent problem in women's health area that can expand from girls to also adult women. We also believe that this immuno-isolating platform can be used as a platform for other cell-based therapies, such as mutable vaccines and in situ production and delivery of proteins and peptides. And we also been able to gain substantial business traction without any dilutive funding. So my ask for today is $15 million Series A to start and accomplish phase 1 clinical trial.
Thank you, Ariella. If we have questions, please enter them into the Q&A function. So here's our first question. Can you please explain what the immunocapsule is made of?
Yes, absolutely. Thank you so much for this question. So immuno-isolating capsule is made from hydrogel. So it's polyethylene glycol hydrogel that has been tested and has been approved for other application for human body, such as ingestion, and also in a lot of cosmetic products. So it's polyethylene glycol hydrogel.
Thank you. Are there any FDA-approved similar capsules for any other types of cancer?
So not that I know of, right? So there is a huge field that's trying to develop immuno-isolating delivery to treat diabetes, right? So to encapsulate islets that can be delivered because it's from donor, so you have to protect it from the host to deliver islets and implant it. However, there is nothing FDA approved yet.
With a tissue delivery, what are the risks of immunogenicity issues? Sorry if I missed that.
Excellent question. So we've tested everything in mice, right? So we did syngeneic, allogeneic implantations in mice. We also tested human tissue in immune compromised and also immune competent mice. And because it's immuno-isolating capsule, cells from the host do not infiltrate in. So there is no immune rejection. We also tested it in sensitized hosts. So we sensitized the mice towards the same allogeneic tissue and then implanted it in mice that already have circulating antibodies, and still the tissue was able to act.
What are your clinical efficacy endpoints?
Excellent. So our phase 1 clinical trial is targeted for adult women with premature ovarian insufficiency. So these women will not have any ovarian function. We anticipate to start measuring hormone production, such as estrogen, progesterone, lowering of FSH, increase in AMH, so all the hormones. And then midway, we will also test for the bone health, cardiovascular health, and so on.
Thank you, Ariella. If I can ask back my leaders in UCLA, University of Michigan, and Yale, if you're still here, and we will wrap up the webinar.
Yeah, I want to thank all the faculty members that took their time, from their busy schedule to speak with us today, and obviously all the entrepreneurs and investors that were on the audience. If you saw something that you liked, please contact either Kelly, Josh, or myself, and we'll be happy to follow up.
Yeah, likewise. Thank you, Amir, for your leadership and pulling this together. Thank you so much for all the faculty presenters. It was great to see your presentations. Ariella, it's wonderful to see, in particular, the progress you've made over the years. So thank you all for tuning in and staying here. And likewise, if you'd like more information, in particular about the University of Michigan Innovation Pipeline, we do put out a quarterly startup pipeline report. Would be happy to add any of today's attendees to that. Feel free to reach out, kbsexton@umich.edu. Thank you, everyone.
And thank you, Marvi, for doing all the heavy lifting behind the scenes.